Restoration Gone Wild. Join us for some truly inspiring stories of caring people from all walks of life working to restore and protect our natural ecosystems. Restoration Gone Wild. Fly with us to the border of Washington State and British Columbia, where efforts are underway in the San Juan Islands to save the Pinto Abalone. This iconic invertebrate used to thrive among the rocky shores and is the only abalone indigenous to Washington State and British Columbia. Pinto abalone are a large marine snail, long been enjoyed as a gourmet delicacy, which may have resulted in an unsustainable demand for them as a species. Scientists have determined in recent years that the Washington State population has plummeted to a point where there are too few in the wild to successfully reproduce, and without human intervention, they may be at risk of becoming locally extinct. The goal of this Washington State Abalone Recovery Program is to rebuild a self-sustaining wild population while ensuring genetic diversity within the species. Although this species is almost lost, it hasn't been forgotten by a core of dedicated individuals.
I'm sitting here with Don Rothus with Washington Department of Fish and Wildlife, and we're here at Port Gamble, which is the location of an abalone nursery. And so tell us what you have discovered about Pinto abalone populations in Washington.
I first realized that we had problems with abalone populations in 1994 when we were doing follow-up surveys of our abalone index sites in the San Juan archipelago. What we've seen in our surveys is a dramatic decline in our populations, and as of 2009 we've seen a roughly 83% decline even after the fact that we've closed the Pinto abalone fishery in 1994. So the mystery is, with a complete closure of the fishery, why have these declines continued? Without any fishing pressure on this population, on this species, we thought we'd see just a slow, gradual recovery of this species. But what we've seen is the exact opposite. We've seen continued declines, and in addition to that, what we've seen is very little recruitment of new juvenile abalone into our population, and a persistence of larger abalone. And so it goes against the fact that, this is continuing because of some sort of poaching or illegal harvest that's continuing. There is some other biological factor that's affecting these abalone populations in Washington State. Some studies suggest that we were harvesting somewhere on the order of 43 to 40,000 abalone per year by sport diving alone. And that number may have been way too high for the Washington abalone stocks. So that's one of the reasons maybe we got ourselves into trouble, that we didn't track abalone sport harvest very well, and once we noticed a decline, maybe that was too late.
What do we do in a case where there are so few abalone out there that they cannot successfully reproduce? What are we doing now?
The University of Washington and Dr. Carolyn Friedman's lab, they've been very supportive at getting us to where we are today with our knowledge of these declines.
One of the things that we're doing here in this lab is collecting wild male and female abalone and crossing one male with one female to produce separate families of abalone. And the reason that we're trying to produce the separate families is to maximize the potential for genetic diversity in the animals that we're producing. When you're working on a restoration project, that your first goal is to do no harm. So that means trying to maintain a genetic integrity of your animals, trying to optimize rearing methods so that they have the best survival in the wild, and also making sure that we're not introducing or moving any disease agents around.
One of the advantages of us having this hatchery facility here in Mukilteo is the relationship that we have with NOAA. They're providing this lab space for us. It's unique because we have tremendous access to large volumes of flow-through seawater, which is really vital for what we're trying to do. Abalone they're high-flow critters, so they like good water quality in high volume. The tanks that we have here are all designed as flow-through tanks. So water is being pumped from offshore here in Mukilteo, comes to the facility and goes through a series of sand filters that NOAA maintains, and then it comes into our lab and goes through an additional series of filtration before it comes into the tanks. And as a biologist, it's a fairly unique opportunity to have such great access to really high quality seawater.
So what we have to do in our restoration work is make sure that we're not breeding just a bunch of brothers and sisters, but instead are breeding a lot of different families and different combinations of genotypes. So we end up with ultimately as diverse a population as what we started with in the San Juans. And right now we have enough wild animals in the San Juans to provide that genetic diversity. In some restoration programs where humans come to the restoration front too late, they end up having genetically poor populations to start with to try and rebuild the wild populations from. And we're hoping, knocking on wood, that the abalone haven't declined that far.
This tank represents one of our natural habitats, and here's an example of a rock that I pulled out and flipped over, and we're comparing this type of a culture method to the adjacent tank in which animals are reared in more conventional methods. We have these PVC tubes that we can place in the tank to hold kelp to provide access to their feed. And again, you can see a number of Pinto Abalone with different colored tags. You can see how quickly they respond to the light and actually try and get into a sheltered location.
We have 10 new broodstock animals that were collected in the San Juan Islands by the Fish and Wildlife dive team.
Broodstock are sexually mature individuals of a species kept for breeding purposes only.
We'll pretty much take our broodstock samples from their range in Washington State. We try to have somewhere between 30 and 40 broodstock animals in the hatchery at a given time. And ideally we would keep those just for one season of spawning work, and then they would be reintroduced. The broodstock that we have in the lab now were from March of 2008. At that point we collected 35 broodstock animals, and of that group we've had 2 mortalities over the course of a year. So our survival rate in the lab is really good, actually. One of our strategies when we're collecting broodstock animals in the wild is we try to avoid taking healthy aggregations. So if we see a number of Pinto Abalone in a group of 3 or 4 or 5 animals, we try to leave that population alone. We don't want to disturb any natural spawning populations that are there, healthy, doing their thing. So we try to take individual animals, which involves a lot more dive time, but in the long run it's worth it to leave those groupings of Pinto Abalone together and taking the single animals that are probably ecologically dead anyway because they're isolated. We're taking maximum shell length because it allows us to see whether or not the animals are growing once they arrive here in the hatchery. That's kind of an indication of health, whether or not they're eating well. You have to be careful with abalone. They do injure easily. Injuries that you cause are slow healing or may not heal at all. They're hemophiliacs. They just don't heal, they'll bleed. And an animal that you injure, you may not see mortality right away, but a month later, you may see that animal die, and you can probably be pretty certain that it's because of the injury. So we try to handle them as little as possible. It's 144. Wet weight is 368 grams. One of the ways that different abalone species are identified is morphometrics. There are some species that look similar to each other. Number of open respiratory pores is one of the identifiers. Pinto Abalone typically have 4 to 6 open pores, and there's other species that may have 15 or 20 open pores. And the tags allow us to track survival of reintroduced broodstock. One of our strategies is use our broodstock population for about a year or so, try to produce as many families as we can from those broodstock, and then we reintroduce those animals to the wild as part of an aggregation. We use 2 different tags. We use a larger disc tag, it's a vinyl tag, and then we use a smaller, what's called a bee tag, and it's actually used for honeybee culture. These bee tags are used to mark the queen bee.
Tag. This is our Floyd tag.
But the bee tags work great for juvenile animals that are 20 millimeters or less, and the vinyl tags work great for the larger adults. Eventually all these animals are going to get reintroduced to the wild. By rotating our broodstock, it keeps genetic diversity high with our hatchery juvenile population, but it also gives those animals a chance to get back out into the wild and maybe do some natural spawning on their own.
These extremely rare photographs show Pinto Abalone spawning in their natural habitat. This is known as broadcast spawning. The success of this method depends on an adequate concentration of males and females in close enough proximity so that once released, the free-floating reproductive cells have a chance of finding each other. This may well be the key to understanding the decline of the Pinto Abalone and any effort to aid in its recovery. In the lab, spawning is stimulated through a very special technique.
We use a hydrogen peroxide protocol. It's commonly used in abalone culturists in Japan and Southern California. It's a 6% hydrogen peroxide solution. They're in the peroxide solution for about 3 hours, at which point we take them out, put them in buckets of fresh seawater, and usually within 2 or 3 hours of that, they start to release gametes. And it's almost like clockwork. As soon as they come out of the peroxide and go into the fresh seawater, oftentimes we see that release of gametes almost immediately. So we keep them separate, hoping that they'll wait to spawn until we ask them to. And then over the course of an entire year of culture here in the lab with the juveniles, we may only see 1% survival. So I may settle out 50,000 animals, and a year later, when we get to the point where we have 10, 15 millimeter juveniles, we only have 1% of what was settled out initially. This tissue is lined with those tentacles that you see when they're in the water. They kind of splay those out, they're sensory organs. And we can actually just clip off 2 or 3 of those tentacles, and that will be enough of a genetic sample. And we use that to tell us who's who.
I was going to say, these things do get domesticated in a sense, the ones we have for a while in the lab. I mean, I'll go into the tank with some algae and I lean over and they can just detect my shadow and they know I'm the guy with kelp. They will literally rear back with their front of their foot and grab the piece of kelp out of my hand, some of them do. So I think that's part of it, is that they get kind of used to a totally different setting than they'll be in the wild.
Yeah, if we can rear them in an environment that's less domesticating, maybe that's a strategy we start using.
We like them. We do. We're passionate about these guys. So.
These marks that you see here on the PVC are grazing marks from the adult Pinto Abalone. They just move back and forth and kind of see these patterns that they make.
We should definitely be concerned about with Pinto Abalone is our populations are really low right now. They're tenuous at best under good conditions. But in our current environment, we're looking at potential issues of ocean acidification. We're looking at changes in water temperature. We're looking at increased sedimentation or siltation. We're looking at some large-scale physical disturbances, as there's propositions for tidal power and some other large-scale projects in the San Juan archipelago and around this area that might affect these abalone to a level that we don't yet understand. And if you have a population that's already barely hanging on, it often doesn't take much to perturb that population just enough to set it past a tipping point, past a point of no return.
We're at the Port Gamble Abalone Grow-Out facility right now. This facility is a collaborative project between the Department of Fish and Wildlife and the Puget Sound Restoration Fund. We've been growing out juvenile abalone here at the lab for close to a year now. These animals are coming up on 2 years old, and they're ready for outplanting to the wild as part of a larger scale restoration project. So this is the outplant tube that we've designed for getting these juvenile abalone out to the wild. It's basically a 6-inch PVC, has oyster netting on each end of it.
Each of these tubes is going to be assigned a certain number of animals, and the animals that will be dumped into each of these tubes represent a cross-section of the families we have here in this hatchery. And the idea is to get a mix of the individual families that we have from this outplanted to each of the individual sites. So the critical job for us as a team today, we double check when we put an animal into a tube that that animal actually belongs in that tube. The first set of 20 tubes that we have here will be taking animals that we have specifically tagged. So you're looking at a tag number and its color, and then looking on a master sheet that we have and saying, okay, that number is appropriate for that tube, that color is appropriate for that tube. The 20 tubes will go into a large fish tote that will be in the back of the red truck. We'll get that truck underway up to Anacortes so the animals are out of nice aquaculture facility for as short a period of time as possible.
So this is a Pycnopodia sea star right here. It's actually a really small one, but this is probably one of the primary predators of Pinto abalone. We like to keep one of these in the lab as a abalone removal tool. It's much easier on the animal than a spatula or some other tool. And they'll respond to this cue fairly quickly. We can just drop the Pycnopodia right down on top of the abalone, and as soon as they sense that, they'll start moving around.
Abalone can move relatively quickly because the left and right side locomotory waves of their feet are in 2 different phases. allowing them to move much more efficiently than other gastropods.
Get me out of here!
You can see their epipodial tentacles start to come out.
There's a lot of variability in size both within and between families. It has a lot to do with the age or size that the abalone weaned from their initial diet of benthic diatoms to macroalgae. And it seems that when they wean from the diatoms to the algae, they really start to take off in terms of growth. My belief is that that timing is what dictates the growth rate of the abalone, and that certainly just happens on an individual basis, seemingly within each family.
Why are we doing this? Right. What is it about the Pinto Abalone that has galvanized a group of people behind its recovery?
All of these species are interconnected, part of the larger ecology of a particular area. For instance, abalone are really important herbivores. In that function, they clear large areas of rocky habitat other important invertebrates can recruit to, and basically help to increase the diversity of animals. The loss of this one species has a cascading effect on the other species that are dependent on those open, clear areas. Particularly important in abalone recovery is the fact that those grazed areas exist, because juvenile abalone rely on these clean, rocky habitats covered with this beautiful pink crustose coralline algae. So in other words, without a significant adult population grazing these areas clean, it's going to be very unlikely that these juveniles, if they do exist, can find suitable areas to settle onto and survive and propagate.
So we're at Shannon Point Marine Center in Anacortes, Washington right now. We've developed a collaboration working with the researchers at Shannon Point and working with their facility, their boats, and their dive teams in order to outplant our abalone. First spawned in captivity in the Mukilteo hatchery. They were then reared in our Port Gamble hatchery, and now they're loaded into tubes. They all arrived last night by trucks, and inside each tube there's about 31 abalone from 5 distinct families. All of these tubes here, 40 tubes, about 1,200 animals, represent the first ever enhancement outplant for Pinto Abalone in Washington State.
Due to the delicate nature of these young abalone, it is essential that every effort is made to minimize stress. The tubes are put into tanks that have continual free-flowing filtered and degassed seawater while waiting to be transported to the outplant boat. This ensures optimal water quality for these fragile animals.
Now we will transport all of these tubes in a number of coolers, constantly changing the water every 30 to 40 minutes. Conveniently, our outplant sites here are really close to the marine lab, so we're hoping that that is really going to help us with the water quality issues and help us to prevent having any problems associated with working with these really fragile animals.
We're doing our initial deployment of the nursery module.
The modules are intended to protect the young abalone in their first few hours out in the wild.
Since we've already figured out where these places are, we drop down and we'll put a line or a tape around the perimeter to give the rest of the team some idea of where they can evenly distribute the modules.
We've chosen 4 different sites for this particular outplant project. We use a basic set of criteria for what we want in our outplant sites for these juvenile abalone. And it's mostly based on habitat. So we're looking for solid rock, boulder type habitat, rocky reef habitat. We like really dynamic, high flow areas. We also look for solid rock and boulder habitat that's covered with crustose coralline algae. It's the pink rock, which indicates good clean habitat, and it's also a settlement cue for abalone. We look at species composition on the site, so we like to see that if there's 1 or 2 adult abalone there, that's a good sign that it's probably a place that the juveniles are going to do well. We can look at the presence and absence of other critters, so we like to see that there's maybe some sea urchins and some sea cucumbers utilizing this habitat. We also take a look at predators on the site. So try to avoid places where there's a real high abundance of sunflower sea stars and red rock crabs and things like that that might predate on young abalone that are newly introduced. We also like to see that there's good macroalgae present, so things like bull kelp and other understory brown algae. We are aiming for sites that are at depths of maybe 25 feet on the deeper end, up to about 10 feet on the shallower edge. And we try to set up our plots so that they're roughly 8 by 10 meters in size. We'll actually mark these sites. Each site will have some pier blocks on each corner with some yellow floating poly lines so that we can easily identify these sites when we come back to do follow-up surveys.
We just put 300 animals down on one of our sites. We had 10 tubes total for the transport. It was just really cool to see the animals, to actually put them in their habitat now that we've been raising them in the lab in the hatchery. And it's just kind of exciting, it made me happy.
Well, we're at our second site now. We just finished our first dive. 4 of us successfully outplan a quarter of our animals. Pretty exciting to see it actually starting to come together. Pretty satisfying coming up at the end of that dive and be able to look down and see those 10 blue PVC tubes, each with 30 animals in it. It's sunny, it's beautiful, it's calm, the current's cooperating. Visibility is not fantastic, but it's better than it could be for here. So, yeah, we're out planting abalone.
It's a big kelp bed, and there's lots of krill in the water today, down along the bottom on top of the rock, so lots of little tiny red krill just moving all over. And so it just looked like the rocks were kind of moving, and just lots of little critters down there. It was kind of fun. Like you looking for a spot to put the tube and you're like trying to move them out of the way, but I just kind of got them excited so they just move all over.
1,200 abalone, we did it. 4 sites, 1,200 abs, 2 years. There we go.
That's perfect.
Success. Now they just have to survive.
Mission accomplished. And so we're all done. We'll come back tomorrow and set them free.
The humble Pinto Abalone will never provide the dramatic images like those of a pod of orcas cruising among the islands, or a bear catching a salmon in mid-flight. It will continue to just quietly go about its business, unseen by nearly everyone, fulfilling its role in a complex ecosystem that we are only now just beginning to understand. But what we do suspect is the number of Pinto Abalone remaining in their natural habitat have declined to a point where without our help and protection, their demise will be another blow to biodiversity and eventually become just another minor footnote in the history of planet Earth. But thanks to dedicated people from across many disciplines, all working together, this scenario doesn't have to be the fate of Pinto Abalone, of our natural world, and ultimately of ourselves as a species.
Do I have any boogers on my face? I feel like I have boogers on my face.